This is a GCSE Combined Science Trilogy Higher Tier Biology Paper 1H. Plants absorb light for photosynthesis, which is the equation for photosynthesis. So remember, with photosynthesis, you're taking carbon dioxide and water and producing glucose and oxygen, which is why it's this one. A student investigated the effect of light intensity on the rate of photosynthesis. Figure 1 shows the apparatus. So remember, those bubbles of gas being produced must be oxygen. This is the method used. Set up the apparatus as shown in figure 1. Place the pondweed 10 centimetres away from the lamp. Switch on the lamp. Record the number of bubbles of gas produced in 5 minutes. Repeat steps 2 to 4 with the pondweed at different distances. So what was the independent variable in this investigation? Well, that is what you're changing, which, remember, they told us before was the light intensity, which they were affecting by changing the distance. So it's actually going to be this first option here, distance of the pondweed from the lamp. If they'd asked you what the dependent variable was, what you're measuring, it would have been this answer here. The lamp gets warm when it's on. This causes the temperature of the water to increase. Explain how an increase in temperature would affect the results of the investigation. There would be more bubbles of oxygen produced in 15 minutes. And why is that? Well, remember, it's enzymes which will be working faster at higher temperatures. Suggest one way the investigation could be improved so the temperature of the water does not increase. What kind of apparatus can you use to keep temperature constant? You can use a hot water bath. Suggest 2 improvements to the investigation so the results would be more valid. Do not refer to controlling the temperature of the water. So what sort of control variables should we use? So use the same lamp or the same power of lamp. Remember, with experiments, it's always worth repeating and calculating a mean. Table 1 shows the results. Calculate the rate of photosynthesis when the pondweed was 40 centimetres away from the lamp. Give the rate of photosynthesis as a number of bubbles of gas produced per minute. So we know that we have 16 bubbles produced in 5 minutes. So in order to find how much was produced per minute, we divide that by 5, which is 3.2. So 3 bubbles of gas were produced per minute. Give one conclusion that can be made from table 1. As the light intensity decreases, so this is low light intensity at this end, it's high up here because the lamp's close to the pondweed. You can see that as the light intensity decreases, the number of bubbles of gas produced decreases, i.e. the rate of photosynthesis decreases. So this is when you talk about the science. Plot the data from table 1 on figure 2, draw a line of best fit. So when we have a distance of 10 centimetres, we have 120 bubbles of gas. 20 and 56. 30 and 31. 40 and 16. 50 and 10. So you want a curved line of best fit passing through. So I've drawn my line of best fit there. Predict the number of bubbles that would be produced in 5 minutes if the pondweed was 60 centimetres from the lamp. That means I need to draw a line up here to where it hits my graph line and then read off on the y-axis. So you can see that that is around 7. Describe how to test a sample of food for protein, starch and sugar. Give the colours that would be seen. Okay, let's take them all one by one so we don't miss anything. So to test for protein, use Biro reagent. Remember that should turn purple in the presence of protein. To test for starch, we use iodine, which turns blue-black in the presence of starch. And then lastly, to test for glucose, use Benedict's solution, heat in a water bath, and it should turn brick red. And by writing all these things, you'll make sure you get your 6 marks. Fermentation in yeast is used in the manufacture of bread and alcoholic drinks. The equation for fermentation is this. Fermentation is an exothermic reaction. What does exothermic mean? It means releases heat energy to the surroundings. A student investigated the effect of temperature on the fermentation in yeast. This is the method used. Mix yeast with a sugar solution in a flask. Pour a layer of oil over the surface of the mixture. Why have they done that? To stop oxygen reaching the yeast. And why is that important? Because the yeast anaerobically respires. Put the flask in a water bath at 2 degrees and leave for 20 minutes. Attach a gas syringe. Record the volume of gas collected every 5 minutes for 30 minutes. Remember that gas is going to be carbon dioxide. After 30 minutes, move the flask to a water bath at 35 degrees. Continue to record the volume of gas collected every 5 minutes. Suggests why a layer of oil was needed on the surface of the mixture. There it is. And I've already provided the answer: to stop oxygen reaching the yeast. Suggests why the mixture was left for 20 minutes before the gas syringe was attached. That's mainly to allow the yeast to reach that 2 degrees temperature. Steps 1 to 4 of the method were repeated at 35 degrees. The volume of gas collected was recorded every 5 minutes for 45 minutes. Table 2 shows the results for both flasks for the first 30 minutes. Table 3 shows the results for the last 15 minutes when both flasks were at 35 degrees. Explain the results from 0 minutes to 45 minutes for the flask that was at 2 degrees and then moved to 35 degrees. Let's look at this set of data. First of all, we have no gas being collected. Why? Because 2 degrees is too cold for the enzymes in the yeast to work. So therefore we have, as I pointed out before, the gas is carbon dioxide, so no carbon dioxide is produced. However, when that temperature increases to 35 degrees, we can see a huge amount of gas being released because those enzymes have become so much more active. Explain the results from 0 minutes to 45 minutes for the flask kept at 35 degrees. So we're looking at this data along here. So we can see that at 35 degrees, we're producing a lot of gas. And why is that? It's because it's the suitable temperature for the yeast to respire at. So that carbon dioxide is being produced rapidly. But then have a look. After 15 minutes, we can see that the rate at which that carbon dioxide is being released has slowed down. So we need to try and work out why. Well, it's because if you refer to this equation, we know that as well as carbon dioxide being made, we've also got ethanol being made, which is an alcohol, which is toxic at high concentration. So it's likely that's going to start killing the yeast. Pathogens are microorganisms that cause disease, Gonorrhea, malaria, and measles are 3 diseases in humans. Draw one line from each disease to the pathogen that causes the disease. Okay, so I think for me the most straightforward one is knowing that malaria is caused by a protist. Measles is caused by a virus. And then finally, gonorrhea, it's not a fungus, it's a bacterium. It's a sexually transmitted disease. Malaria is transmitted by mosquitoes. Male mosquitoes can be sterilised, so they are infertile. The spread of malaria is reduced by releasing sterile mosquitoes into the environment. Explain how releasing sterile mosquitoes reduces the spread of malaria. Because there are fewer breeding mosquitoes. Meaning that there's less reproduction. And because you have less reproduction, means fewer mosquitoes to spread malaria, or the protist that causes malaria. Pathogens also cause diseases in plants. Figure 4 shows rose black spot fungal spore and tobacco mosaic virus. Name the piece of equipment used to view the virus. Well, a virus, I hope you're aware, is absolutely tiny. So you're going to need a microscope, but you need an electron microscope. Look at the size difference, 16 micrometers versus 2.5 times 10 to the minus 7 meters. How many times longer is the fungal spore compared to the virus? So we do need them in the same unit. Because we need them both in meters, or at least in the same unit, this is how we go about it. Divide by 1,000 twice. So effectively you're dividing by 1,000,000. And then because you're comparing the length of the fungal spore with the virus, you have to identify which of these numbers is smallest. The virus length will be smaller. So we do this. And so actually that fungal spore is 64 times longer. Explain why plants infected with tobacco mosaic virus grow slowly. Remember, tobacco mosaic virus, TMV, causes discoloration to leaves, meaning there's less chlorophyll. And less photosynthesis takes place because you have that damage, that lack of chlorophyll. So less glucose made, which is needed for growth. And fewer amino acids will be made. Figure 5 shows the human heart. Remember, the left and right sides are reversed. So here's the left ventricle, left atrium, right ventricle. Which blood vessel transports blood with the highest oxygen concentration into the heart? So because it's full of oxygen, it must be coming into the left side. It's coming from the lungs, which means it's going to be coming in this direction through the left ventricle and out of the aorta. So it's coming into the heart in vessel D. Blood pressure is a measure of the force of the blood against the walls of blood vessels. Which blood vessel transports blood at the highest pressure? You're looking for the aorta here, which comes out of the left ventricle. So that's actually C. What is the correct order for blood flowing through the heart to the lungs? So we've said it comes into the left atrium, down to the left ventricle, out of the aorta, and then back into the vena cava. Now it's been deoxygenated into the right atrium, right ventricle, and then out of B, which is the pulmonary artery. Remember that arteries always take blood away from the heart. So what is the correct order for blood flowing through the heart to the lungs? Well, the last vessel needs to be the pulmonary artery because that's taking blood to the lungs. Pulmonary means lungs. And remember, I was talking about the right side the whole time, which means the blood's coming into that right atrium, being forced into the right ventricle, and then leaves via the pulmonary artery. Every year, thousands of people in the UK have heart attacks. A heart attack is caused when the heart muscle cells do not get enough oxygen, causing cells to die. Statins and stents are two treatments used to reduce the risk of someone having a heart attack. Evaluate the use of statins compared with the use of a stent to reduce the risk of a heart attack. Evaluate means advantages and disadvantages, or pros and cons. You have to give both. So let's start with statins. So advantages of statins. Remember, these are drugs. And so because they're just tablets, it means that they're very easy to take. You don't have to have a big operation. What are they used for? They're used to reduce blood cholesterol, which therefore helps reduce the buildup of fatty materials in the arteries. And remember, it's that obstruction which leads to heart attacks. So effectively, they are good at reducing your risk of a heart attack. But we need to give disadvantages, but they must be taken long term. And the fact that they're a tablet means there's a chance of forgetting to take them. And obviously, if you don't take them, then you won't have any treatment. Now let's talk about the stent. This is a much longer term solution because after all, it's an operation. How does it work? Well, remember that balloon inside a mesh is inflated, helping to open up that blocked artery, meaning again more oxygen can reach heart muscles, meaning less chance of a heart attack. And the great thing about a stent is that it is effective immediately. Straight after that operation, that person will be feeling a lot better. But what about the disadvantages? But there's a risk to the patient because obviously you're having open heart surgery. And there's an increased chance of blood clots and hemorrhaging or bleeding. So we have written an extremely full answer here. Ta-da. Pros and cons for both statins and stents. Many people who survive a heart attack get out of breath easily when they exercise gently. Explain why heart attack survivors get out of breath easily. Well, it already pointed out that there is death of heart muscle cells. So what does that mean? Well, it means that the heart can't contract as powerfully, meaning that less oxygen is pumped per heartbeat. Less oxygen reaches respiring cells, which are all important when you're exercising as they contract. So how does our body have to counterbalance that? Well, if the heart's not doing a good enough job, then you need to get more oxygen into the blood in the first place, so the breathing rate increases. Scientists have developed patches of beating heart cells to repair damaged heart tissue. The patches are placed onto areas of the heart where the cells have died. New cells grow to replace the dead cells. The patches are made using a person's own cells that are converted into stem cells. Explain why stem cells are used to make the patches. It's because those stem cells have the ability to differentiate into the exact type of cell you're after. So, for example, heart muscle cells, which is obviously extremely useful. So they can differentiate or specialize into new types of cell. The scientists could have used human embryonic stem cells to make the patches. Give two advantages of using stem cells made from a person's own cells rather than embryonic stem cells. Firstly, your body is less likely to reject cells from yourself. And then secondly, there's no ethical issues, because remember, you have to use aborted fetuses in order to make embryonic stem cells, or at least that's one source of them. This question is about plant transport systems. Describe how water is transported from the soil to the atmosphere through a plant. So we need an answer that includes talking of roots. How does it get up the stem? Well, in the xylem. And then how does it enter the atmosphere through the stomata in the leaf? So first of all, water enters the root hair cells. Let's be nice and specific here. By what movement? By osmosis, because remember that is the movement of water. Water travels up the stem in the xylem to the leaves, and then water leaves the leaf or exits the leaf by a process called transpiration via the small pores in the leaf called stomata. That's a really full answer there. Dissolved sugars are moved through a plant in the phloem tissue. What is the name of the process that moves dissolved sugars through phloem tissue? You need to learn that. It is translocation. That means the movement of sugars in a plant from source to sink. Phloem tissue is made of sieve tube cells and companion cells. So the sugar moves in both directions inside that phloem. Remember, mitochondria are there to carry out respiration, to release energy. Explain one way the sieve tube cells are specialised for their function. Just have a look. You can see these holes. They're called pores, and they enable dissolved sugars to move from cell to cell. You could also mention the fact that they have no nucleus or few organelles to maximise space for movement of dissolved sugars. What does the structure of the companion cells suggest about the process that moves dissolved sugars through the phloem tissue? Give a reason for your answer. Okay, remember here that there's all those mitochondria. So why are they there? To enable that movement of sugar by active transport, because there must be a requirement of energy here. So many mitochondria in companion cells. Let's say what they do. Release the energy needed to move sugar in the phloem by active transport. Cramming our answer with marking points, specialised terms. Describe why it is important that dissolved sugars are moved both upwards and downwards in a plant. So we know that the sugars are made in the process of photosynthesis in the leaf, but where do those sugars need to be used? They need to be transported for storage, such as when they're converted to starch, and the sugars actually need to be used themselves in the process of aerobic respiration. So that's why it's important that they can move to where they're needed. So sugars are made in the leaves by photosynthesis and transported downwards to be stored, e.g. as starch. And let's give an example of them moving upwards to growing regions where they can be used in respiration.